Well, hello everyone, and welcome to today's webinar entitled HPAI and its Impact on Food Production Industries. I'm Chris Boomsma, CEO here at the Council for Agricultural Science and Technology, or CAST, and I'm happy to get things kicked off before I hand it off to our moderator and then the presenters. So with that, let's get started and get rolling because we've got a lot of great folks with us today to deliver information on this important topic. First of all, very briefly, CAST is a 501c3 membership-supported nonprofit organization formed in 1972, and in 2022, we celebrated 50 years of existence. We're proudly nonpartisan, apolitical, unbiased, and science-based, very devoted to maintaining that way of operating as an organization. Our membership is both at the individual and the organizational level. At the organizational level, we're composed of 27 scientific societies, 20 universities, 19 libraries, a slew of nonprofits and companies, and then a number of individuals from a variety of states and countries. The mission here at CAST is to convene and coordinate networks of experts to assemble, interpret, and communicate credible, unbiased, science-based information to a variety of audiences, including policymakers, government officials, the media, the private sector, the public, fellow scientists, and the like. How do we accomplish this mission? Largely through the help of many volunteer contributors. And you're going to hear from some great volunteer contributors today. We have board members, a variety of active task force members, authors, paper reviewers, and the like. And we're very, very grateful for these volunteers. As I said, an example is today. Without our great volunteer network, these presentations, these webinars could not happen. So thank you to our volunteer network. With that, I'm going to hand it over to our moderator, Julia Herman from National Cattlemen's Beef Association. Julia is going to introduce our presenters for today, also serve as our moderator and guide this discussion. Julia, thank you very much. And when you're ready, I'm happy to advance slides until we get to our presenters. Thank you.
Thank you, Chris. Welcome, everybody. We're really excited to have this panel of experts on to discuss highly pathogenic avian influenza with us. I am the beef cattle specialist veterinarian for the National Cattlemen's Beef Association. My main focus is on education and outreach to producers, veterinarians, and all the different groups that work together. So I think this will be a great learning tool for me, but also hopefully for everybody in the audience. So I'm going to read all the bios of all of our experts, and then we will get rolling. So first off, Dr. Carol Cardona is the Pomeroy Chair in Avian Health for the College of Veterinary Medicine. In addition to her expertise in avian influenza, Dr. Cardona is interested in zoonoses, infectious diseases that are transmitted between animals and humans, and the role that poultry play in human health and well-being. She also serves as co-director, co-principal investigator of the Secure Food System at the University of Minnesota. Dr. Marie Culhane is a veterinarian and researcher at the University of Minnesota as a professor of veterinary population medicine. Her research involves the antigenic and genetic characterization of animal influenza A viruses, pathogenesis of unique influenza A virus infections in animals, and the possible impact these viruses may have on current vaccination and control protocols for U.S. animal agriculture. Dr. Mo El-Ghazar is a poultry diagnostician and outreach veterinarian at Iowa State University at the Veterinary Diagnostic Laboratory. His research is focused on molecular epidemiology and infectious diseases, identification and characterization within the poultry industry. Dr. El-Ghazar was also a volunteer on our animal work group here at CAST. Next up, Dr. Jamie Jonker is National Milk Producers Federation's Chief Science Officer and Vice President of Sustainability and Scientific Affairs. Jonker is NMPF's lead scientific expert on dairy issues ranging from animal health and sustainability to dairy farm biosecurity, air and water quality, and more. He leads engagement with scientific groups across the country and internationally. Prior to joining NMPF, he worked at the National Academy of Sciences, U.S. EPA, and the U.S. House of Representatives Committee on Ag. Finally, we have Dr. Julie Gauthier, who is the Executive Director of Field Operations for USDA APHIS, or Animal Plant and Health Inspection Service, Veterinary Services. Her DVM is from Michigan State University, and her Master's of Public Health is from Yale School of Public Health. She joined USDA in 2002 as a field veterinary medical officer investigating animal disease outbreaks in southeastern Florida. She's expanded her role in the outbreak and foreign animal disease preparedness realm, and she currently is responsible for policy development for the 2024 H5N1 dairy cattle outbreak. So, Dr. Cardona, I will pass it on to you.
Thank you so much, and I'll get my slides going here and get started. So today I'm going to talk about influenza A viruses in their reservoir hosts, the migration, how the behavior of those hosts and the viruses kind of come together to create a biology, and the transition period that we're now in. So influenza A viruses originate from avian reservoirs. We're going to talk about the nature of birds and their behaviors and how we perceive that, which tends to drive our understanding of influenza A viruses. We're then going to talk about this big shift in influenza biology caused by goose Guangdong lineage H5 viruses in wild birds, and how we understand what's going on or how we don't. And the state of avian infection today. So influenza A viruses are hosted by wild aquatic birds. They're the hosts of the genetic diversity of influenza A viruses. So they host 16 HA and 9 NA types. Those are called subtypes. So when they come together, the H and the N come together to form a subtype. So H1N1 all the way up to H16N9. And not all of them have been detected in wild aquatic birds, but 104 different subtypes have been. And then that trickles down into our domestic poultry. And today we're going to talk about goose Guangdong lineage H5NX, which is our concern today. So I just wanted to show you this life map that maps out where the hosts of influenza A viruses are. This includes mammals and birds. And if we focus in just on the birds, you know, that previous map looks pretty small, but it's not small. So with the detection in blue-footed boobies and red-footed boobies in South America and in ostriches, that now means that we probably have an infection that can be seen in any type of bird. So 9,000 plus species worldwide, including the passerines with the detection in grackles, and columbiformes, with the detection in pigeons with this most recent outbreak. So that's a wide diversity of hosts that can be infected with influenza A viruses, and I'm not even talking about the mammals. So the biology of birds and the biology of those hosts influences how they host influenza A viruses. So first, migration is a critical avian behavior for influenza ecology. Groups during migration are deconstructed and reconstructed. So if we think of groups coming together for breeding, which is immediately prior to when they make their southern migrations in North America, those groups are going to be deconstructed. So those breeding groups are sort of a small bubble. They've transmitted influenza around. And as they're deconstructed and they get reconstructed, like you see in this picture, where you get multiple species sharing an aquatic habitat. You know, you can see some redhead ducks, some scaup, and some coots here, and maybe some blackhead ducks as well. And each of those bubbles is going to come together and bring their own influenza to this new ecosystem of, and those viruses will begin to mix and be shared. Next part of migration that's critical is global movement. So most of you are familiar with this north to south and south to north movement of migratory birds along flyways. But it's important to remember there are a lot of species of birds and of course mammals that are also hosts of influenza A viruses that are not moving. So this creates a dynamic and an ecology where we've got a mixture of genetics and shedding animals, and then going north to south, laid over resident animals. In addition, influenza viruses like water, and so habitats that are linked to water create better reservoirs. So our friend the mallard in the bottommost corner here, she's showing you how she likes to nest on the water. She lives in the water, loves the water, and that creates an optimal situation for influenza A viruses. So if there's a bank, and if you wanted to rob a bank, you would go to sampling mallards. That's where the viruses are going to be found. In contrast, our friend the wood duck here, now he has some influenza viruses, but he has fewer and less diversity because he and his mate like to nest in trees and they like to roost in trees. So they have a less aquatic habitat than our friend the mallard. And then finally, pathogenicity or virulence is going to vary by species, and that's going to influence how how birds mold influenza viruses. So our friend the mallard here may show few or no clinical signs when infected with the same virus that can cause death in our beautiful white leghorn pullets over here. Okay. So new viruses, influenza A viruses have some biological characteristics that we need to remember. New influenza viruses tend to fall apart, especially in the reservoir host, so especially in wild birds. So in wild birds where we have multiple viral subtypes coming together, we tend to see those 8 genetic segments. They're white in the H6N1 and black in the H5N2. They sort of shuffle together like 2 decks of cards, and you come out with a mixture of both the genetic segments, but also those genetic segments are going to imbue both new subtypes, and also new phenotypes. So if we look at influenza A virus in the reservoir in this beautiful study done at Ohio State University over many years, we can see they collected samples along the Mississippi flyway as the birds traveled south from Canada down into the Delta. And so what they saw was that influenza A viruses, the 8 genetic segments are shown along the x-axis, and along the y-axis we can see either days or distance. We can see that the 8 segments stay together for a very short period of time in the flyway. In fact, after only less than 200 days, well, let's see, about 200 days, we see that only 2 of the genetic segments have stuck together. Similarly, across a very short distance do those 8 segments stay together, but each segment may make it the entire length of the flyway. So the segments are recovered, but part of the way that influenza changes its genetics in the reservoir is by reassortment. So now we have to talk about how this new virus, new to us, emerged first in 1996 and has changed the global ecology of avian influenza. It's a virus called goose guangdong 1996, and I'm going to refer to it as goose guangdong lineage viruses. So in 1996 and the decades following, it emerged quickly to take over and became endemic in wild birds in Asia, It then in 2005 moved across the Ural Mountains and into Europe and became endemic in wild birds there. In 2015, we saw an invasion along the West Coast that quickly made its way to the Midwest, but we didn't see it become endemic in wild birds. But in 2022, we've seen multiple introductions of the virus, and I think we can agree that it's endemic in our wild bird population as well. So this has changed our ecology. And so we have to look at some of the things that are happening and do the same things happen with goose guangdong viruses that happen with our North American viruses. And, this study demonstrates that goose guangdong viruses reassort, but at a lower rate than our North American viruses. And then we can see that those reassortants do spread, along flyways and within and by migratory birds, but not potentially as efficiently as our North American viruses. So what is the state of avian infection today? Well, in spring, there's very little surveillance. So we have a few ideas about that. So one way we can see, this is the Raptor Center from the University of Minnesota, and they did a lot of testing during the 2022 outbreak, and we could see it really closely mirrored what we saw in poultry. But today, we're not seeing any infections in those birds. So this source of, likely those birds are those apex predators have antibodies that are protecting them, and we're not seeing cases. We do see passive surveillance that's working. HPAI, and this is the location I got this chart. Morbidity and mortality events, like caught this little sanderling with an H5N1. But I just want to point out this little brown job here, the house sparrow. They have been rarely infected and rarely demonstrated as infected, one in 2022, but in increasing numbers recently. And I'm not sure if this is because of increased harvest being done by studies by Fish and Wildlife Services on positive prems, or because we're actually seeing an increase in prevalence. But that's a ubiquitous species, of course, for agriculture. So this winter and spring, there are likely been changes that may be reflected in what we see in the fall surveillance in wild birds, both the cattle source of influenza virus and the cattle expansions, and the infection of mice, obviously a new food source for many of our birds. So today, we've talked a little bit about how we perceive birds as a singular class Aves, but they're really multiple, and their collective biology shapes the ecology of influenza viruses. The goose guangdong lineage of H5 viruses is unusual, but it's following the same rules. We don't really understand the influenza virus in the reservoir as it relates to our North American strains of influenza, and thus it's hard to understand the transition to goose guangdong endemicity in North American wild birds today. And we don't know much about what's happening in wild birds right now, but we believe this is a period of transition. Thank you for your time.
Thank you, Dr. Cardova, for starting us off. Next up, we'll have Dr. Culhane.
All right. Thanks, everybody. I'll get started. Dr. Cardona introduced us to influenza virus as a problem of avians, and I'm here to tell you about this is a shared problem for avians and mammals, not just our domestic animals, but ourselves. And I'll be brief, but I want to put us all on the same page here, is that influenza viruses, they're part of this family of viruses called the orthomyxoviruses. And I don't want to make anybody on this call a virology expert, but I want to get everybody on the same page. What everyone needs to understand is that the orthomyxovirus family only includes influenza viruses, well, primarily includes influenza viruses. And in influenza viruses, there's 4 major ones: influenza A, influenza B, influenza C, and influenza D. And you need to know that there's several different types of influenza viruses, and that you also need to know that they cause diseases of varying severity. Sometimes it's a subclinical disease, meaning that the animal or bird or human doesn't show any clinical signs, but they're shedding the virus, they're infected and shedding. And sometimes that virus is lethal, like happens with our domestic poultry. The other thing I like to tell you about viruses is that they're negative stranded RNA viruses. And that might not mean much to anybody, but what it means as a virologist is that this virus likes to change and it likes to mutate. All right, so we know that there's several different types of viruses. We know that it likes to change and it likes to mutate. The rest of this talk and everybody else today is going to talk about only influenza A viruses, just like Dr. Cardona did. And the other thing I want you to know is this pretty picture of the influenza viruses on the left shows that there's many segments of influenza virus, and they encode for a bunch of different proteins. And what you need to know about that is that means that this virus is capable of mixing it up and changing its genome and reassorting, like Dr. Cardona pointed out. And that's how it does it, through that segmented genome. But it produces all these different proteins, and those proteins help the virus infect the host and to replicate. And then there's that surface of the virus. We have the hemagglutinin sticking out. That's HA. We abbreviate that. There's 19 different HAs out there. And there's the neuraminidase, the other protein sticking off the surface of the virus. There's 11 different neuraminases out there. And the hemagglutinin helps the virus bind to the host, and the neuraminidase protein helps the virus get out and spread. So now everybody knows a little bit about virology. If you really want to dig in to this topic that I'm going to present today, there's a really nice paper that came out talking about the different host species who is sharing these influenza viruses. And so that's in Nature Reviews Microbiology. It's a nice, succinct, short read, and it's publicly available for anybody to get at. And from that paper, they highlighted that influenza viruses are shared. And this is all those 19 influenza viruses. This isn't even up to date. It should be H19 for the bats. But all these different hosts, pick a host on there, are capable of being infected with influenza viruses. And influenza A viruses, like I said, are shared, but so are influenza B, C, and D viruses. So this is a shared problem that we're dealing with on this planet. But there's some new species since this paper came out that need to be mentioned. And this is where we really got into the shared problem this year in the United States in 2024. In January 2024, a goat living on a premises where there was infected poultry became sick and infected and had abortions due to H5 avian influenza virus. A new host species was found. But then the real big problem, in my opinion, started to happen is that dairy cows became infected with H5 influenza viruses. And even since then, alpacas have been infected with avian influenza viruses of the H5 subtype. So influenza viruses are shared. And hopefully that just these little few pictures has convinced everybody of that. And these shared influenza viruses are concerning. And another really good paper here that just came out adds in these new infected farm animal species. They have the goats, they have the cows on here. They're missing the alpaca, unfortunately. But it shows that humans and birds and our pets and our companion animals, zoo animals, wild animals, farm animals, and the animals of the seas are all infected with influenza A viruses, or can be. And it's just a matter of time before another change happens. So how do we know that influenza A viruses are shared? You can read the papers and find out more. You can believe what I show you. But we know that influenza viruses are shared when people share data regarding the influenza viruses. So we share data about the host that was infected with influenza viruses. Was it an alpaca? Was it a chicken? Was it a laying chicken? Was it a broiler chicken? Was it a meat turkey? Was it a breeder turkey? We share information about the host. We share the location so people can say, hey, that's never happened before there, or we don't know that it was in that country before this time. And so that's another reason why we have to know time of detection, and we share that information. And somebody will say, well, that happened a long time ago, and now it's happening again. So we need to share time of infection to know that influenza viruses are shared. And then we also need to know how that animal or human or bird got infected by sharing what happened and who they had contact with before, during, and after infection. And then in the laboratory, we start to share things like what we call virus phenotype, or what are the characteristics of this virus? What is it doing? How much disease is it causing? Where is it binding to? Is it replicating in the eye? Is it replicating in the urinary tract? Is it replicating in the lungs only? Where is it in the tissues and cells of the host? And then we share information about the virus genotype. What is the genetic makeup of the virus? What is its viral RNA that is making it unique to that influenza virus? What is it made of? The virus genotyping really helps us understand how influenza viruses are shared. And there's another free publication out there, out in preprint, a preliminary report on the genetic data. And there's also published data or preprint data from a variety of different government agencies as well. But this schematic from this publication here that's freely available online shows the phylogenetic, the genetic relationship on a tree, like a family tree, that shows the relationships between the HA segment, the hemagglutinin segment of the virus, in different host species, and when our current problem with H5N1 likely spilled over into cattle. So they are able to, virus researchers are able to look at the genetic information and combine it with the date and location to find out when did a wild bird share its influenza virus with the ecosystem that a dairy cow was in. And it looks like that might have happened back in November. And then the outbreak really got going in January. And then are humans being infected? And where did this virus come from? Did it come from wild animals or other different wild animals? That's what the virus genotyping, that shared information, tells us about this shared virus. And that gets us into the current situation. When we look at bovines, they haven't been in any of those pictures of late. But if we look back, and this was a nice presentation that was from April 2024, 1949 was the first recorded evidence of influenza virus in cattle. And then the first virus isolated from cattle happened 2 decades later. And in the 70s and 80s, people were describing illnesses, either respiratory or milk drop, in cattle in different countries like Russia and England. And then the 2010s, this was the main focus, is this influenza D viruses seemed to be the primary host was cattle. And so we were focusing in the 2010s and 2015s of influenza C and influenza D viruses in cattle, not really looking at them for influenza A viruses. Haven't seen that since the 60s, 70s, or 80s. But that brings us to today, the 2024 situation, where we have this H5N1 influenza virus isolated from cattle with milk drop. And you're going to hear more about that just after I'm done. But time goes on. We're not going to stop in 2024, and influenza viruses are going to continue to be shared. And what I want to remind you, and Dr. Cardona hinted at that, is that in 2015, the H5 influenza outbreak in U.S. poultry was considered the largest foreign animal disease outbreak in U.S. history. But what's going on now since 2022 and went on in 2023 and now in 2024, we still have H5 outbreaks occurring in U.S. poultry and now it's U.S. cattle. But so much has been learned about disease control that as time goes on, we'll be able to understand this virus and how we can protect our animals even more. With that, I thank you for your time, and I'll pass it over to the next speaker.
Thank you, Dr. Culhane. Such a complex problem, and I really enjoyed the graphics that really brought a little bit of clarity to what we're talking about today. Next up, Dr. Alcazar.
Hello, everyone. I hope you can hear me speak. If not, just let me know. Thank you, Julia. Thank you, Megan and Chris and Cass for putting together this webinar and for inviting me to speak. So I'm going to switch gears a little bit. I'm not going to be talking from the virology point of view. I'm going to be talking to you from an infectious disease prevention, control, and eradication or elimination point of view. So these are the lessons we learned through our outbreaks of highly pathogenic avian influenza in poultry. So just to put everybody on the same page, these are the different steps from detection to elimination of avian influenza in poultry. So we detect the virus, we know there's a positive case, that's an index case. We draw a circle around that case, that's what we call the control zone. We conduct surveillance in that control zone to make sure that there are no more cases in close proximity to that index case. And then we proceed to depopulate and dispose of the index case, clean, disinfect the site in the step we call virus elimination, have some downtime to make sure the virus is completely eliminated from the site, retest after the downtime. And if there is no positives throughout these different steps, we release the control zone and we consider the virus eliminated from that specific zone. So just to give you a visual presentation of what a control zone in a highly pathogenic outbreak looks like, this was the map of Iowa in the 2015 outbreak. And you can see the yellow spot, that's the index case surrounded by a control zone in which commercial poultry and other poultry is tested for surveillance. And once everything is negative, these controls are released. And you can see some of them are conjoining to make some odd shape, but initially these are just circles of 10 kilometers. So that was 2015. And I put the map of 2015 outbreak, county by county positive, versus the map of the 22-24 outbreak that is extending until today in poultry. And by a quick glimpse, you can definitely realize that the 22-24 outbreak is a much bigger outbreak compared to the 2015 outbreak. However, I will say that our prevention, control, and eradication or elimination in the 24 outbreak was much more successful compared to 2015. And you might ask yourself, how is that possible? And I will explain to you that this is my conclusion is due to the fact that in 2015, the prevalence of the H5 influenza virus in the wild bird, which is the source of introduction to domestic poultry, was very low, or relatively low. So that incoming load of the virus, the pressure, if you wish, on the poultry industry was low. And the size of the outbreak in 2015, which was just mentioned that at that time was considered the largest foreign animal disease outbreak in the United States, the size of that outbreak resulted primarily, the majority of these cases were horizontally transmitted from commercial to commercial. And we can tell, as was explained here in a little bit, by looking at the sequence, we can say, yeah, that's a horizontal transmission, that's commercial to commercial, and that is not an introduction from wild birds to poultry. On the other hand, I say 22-24 outbreak was much more successful because of two reasons. The incoming pressure, the prevalence in wild bird was much higher compared to the 2015 outbreak. Not only the diversity of geographical locations where these viruses were picked from wild birds, but also the diversity of species and the number of cases in wild birds. So the incoming challenge was much higher in 22-24 outbreak. However, our success was that we very much stopped or significantly decreased the number of horizontal transmissions that happen industry to industry. And the majority of cases in the 22-24 outbreak happened because of initial introductions from wild birds to domestic poultry. So if that's the case, there are lessons that we learned in 2015 that we then took and implemented towards the 22-24 outbreak control and elimination that allowed for such success. And what are these lessons? So again, these are the steps from detection to eliminations. And we quickly realized from 2015 outbreak that early detection, early depopulation, and early disposal are the key steps that would allow us for much better prevention, control, and elimination of highly pathogenic avian influenza in domestic poultry. Now, you might ask yourself, okay, wait a minute, what about biosecurity? You're just talking about detection, depopulation, and disposal in domestic poultry. I thought biosecurity is important. Biosecurity is very important. Biosecurity is our first line of defense, and probably the most important line of defense against infectious disease, whether that's in poultry or cattle or any other population. However, microbes move in ways that we don't intuitively understand. It is hard for us humans to understand how these microbes move between different hosts and between different species. Therefore, we know that there are weak links in biosecurity, and as the image shows here, your chain is as strong as your weakest link. We prepare for these weak links, these gaps, in biosecurity, which allows us to formulate a second line of defense, a third line of defense after initial biosecurity line of defense. And that's what we learned in 2015. It is the early detection, early depopulation, early disposal that allowed us to formulate the second and third line of defenses so that we can effectively break that cycle of infection and horizontal transmission from commercial poultry to commercial poultry. So these are the 3 lessons we learned from 2015 that allowed us a much successful control in 22-24 outbreak. Now, are there any of these lessons that can be applied towards the current outbreak in dairy cattle? And very quickly, I will tell you that depopulation and disposals are not it. However, early detection is still a lesson that we learned in poultry that could very much be applied in the current outbreak in cattle. So what does that early protection looks like, or early detection looks like in poultry? So we are fortunate in the poultry industry that we have been expecting such outbreaks for a very, very long time, for decades, as Dr. Cardona mentioned, the history of this disease. So we have a program in place. And that program in place looks like that. We have a very strong, very sensitive, very specific test that we use for detection. But not only the test that is run in the lab, before we run the test, we know what type of samples we collect. It's oropharyngeal swabs in turkeys and chickens, but it's fecal or coaxial swabs in ducks. The number of swabs we collect, the frequency and the time, and the supplies. We have supplies, brochures, and training programs in place. So early detection program in place is more than just running a test. There are a lot more logistics involved in such an early detection program. Now, can we say that any future detection program in cattle can learn from these steps? Do we have anything in place that allows us to implement such early detection program in cattle? And the test is the same. So we can use the same test to test for avian influenza in poultry, but it's the same test that can be used in cattle. The PCR test itself is the same. However, the type of samples, the number of samples, the frequency, the supplies and training, these are all parts of the program that need to be implemented so that we know the right sample for cattle, the right frequency for testing in cattle, et cetera, et cetera. So this is going to be my final slide here. My recommendation is let's try and implement an early detection program in the cattle population. And the simplest way to look at this is to test before we move. The cattle population moves around the country, and that's how we know that the virus is transmitted. You just want to emphasize, it is not a continuous spillover from wild birds to cattle that is happening over and over and over again. We know from the genetic sequence that that spillover event most likely happened just once, and that spread of the disease between the cattle or within the cattle population happens because of the horizontal transmission cattle to cattle. So if we test before we move, we know that we're moving animals that are not shedding the virus. implementing an early detection program will help us define and reduce the prevalence of the disease in the cattle population, which in terms will help us implement more effectively a biosecurity program and help us more effectively break that cycle of infection from cattle to cattle. And only then, if we have a successful early detection program and we define and reduce prevalence, only then elimination will become feasible. And I know that the dairy industry is very different from the poultry industry, and there are many, many challenges facing that potential early detection program. But I think still early detection and the goal of elimination of highly pathogenic avian influenza in cattle is still a worthy goal to pursue. So with that, I want to thank CAST again, and thank you for your attention, and looking forward for questions and discussions if we have some.
Thank you very much. Bringing in that practical piece on what producers can be doing on the farm. I think that's great. As I mentioned earlier, Dr. Alcazar was a volunteer on our animal work group. He rotated off. Our next presenter, Dr. Jonker, is also on our animal work group and has brought a lot of his experience onto our group and working on some different issues. But next up, let's go, Dr.
Great. Thank you, Julia. I am Jamie Jonker, the Chief Science Officer with the National Milk Producers Federation. And I'm going to talk to you about what has been now 90 days in the U.S. dairy sector. The first confirmation of H5N1 in dairy cattle occurred on March 25th, so just 3 months ago. As of June 21st, there are now 121 confirmed herds in 12 states. And as you can see on the graphic on the right, provided by USDA, in the last 30 days, there have been 58 dairy farms in 8 states. And you can see from the map the intensity of the blue color as to which areas are more hot right now, that being Idaho and Colorado. And this is new in the dairy sector. While you saw information about influenza being in dairy cattle first being reported in 1949, this is an unexpected turn of H5N1 being in lactating dairy cows. And one of the interesting things about H5N1 in dairy cattle is that it is going into the mammary gland rather than manifesting itself as a respiratory issue. So what do we see when a dairy cow becomes infected with H5N1 virus? These are the symptoms that we would see in a clinically infected cow. We would have, occur in lactating dairy cows of all ages and stages of lactation, although they are more likely to occur in multiparous cows that are cows second lactation or older, and in mid-lactation being around 150 days or so. On average, while this does vary from farm to farm, 10 to 15% of lactating cows do have clinical symptoms. And unlike in poultry, where it's a high mortality event, it's a low mortality event in dairy cows so far, being in the 1 to 2% range. What do we see in dairy cows? We get a drop in feed intake, which decreases their rumination. They also have a drop in water intake, which leads to dehydration. These symptoms really come about because of what happens in the mammary gland. We have significant levels of virus that are in the milk, and milk yield in these clinically infected animals drops precipitously. Interestingly, in some cows that are severely affected, all 4 quarters of the mammary gland are infected. However, in some cows that can be clinical, but are less severely infected, may only have 1 or 2 quarters infected. When a dairy farm gets this virus into their herd, the general timeline is from the first time that they see clinically infected animals, it peaks in the dairy herd around 1 week in, and then tapers off at about 2 weeks in. So they're at their peak about 1 week in where they have 10% or more of their animals that are having to be removed from the general herd population and taken care of in their hospital pen. Because of the volume of animals, most dairy farms do not have hospital areas that are large enough to accommodate this many animals during a health outbreak. They're actually dedicating a pen within the general barn for this. We find that milk production recovers at about 30 to 45 days later, but not all cows that were clinically infected recover in milk production. Some do not recover milk production at all. And so where did it come from? We've had a little bit about this earlier from earlier presentations, but there was this reassortment that led us to this H5N1 genotype B3.13 that came about late last year. Sometime probably in the last month of last year, it came through an unknown host into dairy cattle. It circulated for about another 6 weeks or so, and in the early part of February, late part of January, we actually started seeing a mystery disease in the Texas Panhandle. Because we were not expecting high path AI H5N1 in dairy cattle, it took until March 25th to actually diagnose this as the cause. Subsequently, within a herd, because it's in the mammary gland, there is some spread that happens during the milking process. We have also seen spread from one herd to another herd due to the movement of infected animals, animals that may be infected but do not yet have clinical symptoms. We have also seen lateral flow from dairy farms to egg laying and turkey operations. Through the epidemiological investigations that USDA have been conducting and are still ongoing, the likely routes of travel between those dairy farms and those affected poultry farms may include such things as people, including in Michigan, where there were in fact some shared workers who worked on dairy farms and on poultry farms, and through mechanical conveyances through things like vehicles. And that does get us to where there has been that lateral flow of the dairy strain to egg-laying operations and turkey operations in at least 4 states. And we know that it's the same dairy strain due to the whole genomic sequencing that's been happening across the many samples that have been submitted to our non-laboratory processes. One thing that we do know, based upon the genetic work, is that there is not a lot of change that is happening in the virus so far as it's come through the dairy farms. Through the genetic sequencing that's been done so far, roughly 99.94% of the genes are the same across all of the samples that have had whole genomic sequencing so far. But as we heard from Mo as our last speaker, one of the ways that we need to work to contain and then eliminate the virus is to look at ways in which it is spreading and then take action to reduce the risk of it spreading through those mechanisms. One of those is the movement of lactating dairy cows. And here we can see the requirements that came out from USDA in its federal order at the end of April, which is the requirement of testing of lactating dairy cattle prior to the interstate movement and submitting those into the non-laboratory network. There has been quite a bit of USDA support, which has been announced for both affected dairy premises, those 121 that have been identified through last Friday, and also some additional funds that are available to any dairy farmer in this process. And the one I would point out here for that is available for all dairy farmers is that biosecurity planning to help them put in plans and then work on biosecurity implementation. As we've heard, that biosecurity is a part of the process in helping to minimize the spread of this issue. So we know that the H5N1 dairy strain really likes the mammary gland. There are receptors in the mammary gland where it replicates very well. We have seen some positive samples in urine and some positive samples from nasal swabs, but it seems to settle into the mammary gland. This is not to say that there's not some spread which could occur from cow to cow through an aerosolization process, particularly in the holding pen prior to milking in the parlor. But clearly the high levels of virus that are in the milk for clinically infected animals suggests that one route of spread within a dairy farm from cow to cow is through the milking process. And in fact, it seems to be acting in dairy cows a lot like a mastitis rather than a respiratory issue. And this gets us to focusing on biosecurity. So we look at kind of creating a moat around the dairy farm, the line of separation, and this comes from the Farm Biosecurity Program and the Secure Milk Supply Program, which is looking at ways to minimize the introduction of disease into the farm. And then for those farms that have had an outbreak of the H5N1 dairy strain, to minimize the potential for it moving from an infected farm to a non-affected farm. We have in the U.S. Dairy industry the National Dairy Farm Program, Farmers Assuring Responsible Management, which is our on-farm quality assurance program. The largest part of that program is our animal care program, with more than 99% of dairy farms in the U.S. are involved in that. And biosecurity is actually just a unique part of that animal care program. We have elements within that program for everyday biosecurity, but as we have learned as this outbreak continues, we are actually suggesting that dairy farmers implement some elements of enhanced biosecurity, which in our program are aligned with the requirements under the Secure Milk Supply Program in the event of a foot and mouth disease outbreak. Now, it's not every single element that would be required if there was an FMD outbreak in the US, but certainly several of the elements that are being recommended today are parts of those enhanced biosecurity plans. I did want to briefly mention that we also need to look at the potential impacts on our dairy workers. And to date, there have been 3 dairy workers that have had high path AI H5N1 impact them. CDC is now monitoring over 500 dairy workers. And in this case, the first 2 that were infected had been in contact with clinically infected cows. They did not wear eye protection. They had conjunctivitis, and you can see on the right-hand side of the slide the inflammation in the eye. There are actually receptors in the human eye that this virus can attach to and stimulate a response. In those 2 cases, while they had the eye issue, they did not have any respiratory issues. In the 3rd case, the most recent case, this dairy worker also was in direct contact with clinically infected cows. This person did not have conjunctivitis, but did have respiratory symptoms. And again, this is the same genetic strain that's been found in the dairy cows and poultry farms most recently infected. So this is where we get to the point of what should we be doing to help protect our workers. It's protecting the mucosa of the eyes, the nose, and the mouth, so that we are limiting the possibility of virus entering those areas. You can see the person here on the right-hand side wearing the eye goggles. For those 2 workers that had conjunctivitis in the milking parlor, you can incidentally get milk on your hands and rub your eyes, accidentally, potentially introducing virus into that mucosa. We would also look to further protect the nose and mouth mucosa as well. Protections, particularly for those that are involved in sick or dead birds or other animals on the farm, or those that are directly dealing with clinically infected dairy cows. I'm going to mention just very briefly that the U.S. milk supply goes through what is called the Grade A Pasteurized Milk Ordinance, implemented by the federal government in cooperation with state regulatory authorities and the industry. FDA has conducted a retail survey where they did find positive PCR returns indicating viral fragments in those fluid milk, cottage cheese, and sour cream dairy samples. But importantly, no live virus was detected. FDA also has done a survey of retail powdered infant and toddler formula. All of those were PCR negative, indicating no viral fragments whatsoever in those. FDA is in the process of continuing to do pasteurization research, but we feel confident that pasteurization works as we know it does, which is to reduce bacterial and viral loads in our milk supplies, making them safe for consumers. The World Organization for Animal Health has issued some HPAI guidance in cattle. There is the link at the bottom there that we will share out afterwards. But I think importantly, this is a reportable disease, and the U.S. government has reported the outbreak in cattle to the World Organization for Animal Health. In doing so, it has been able to share ongoing information with our trading partners, and to date, we have not seen any disruptions in the trade of dairy products from the United States to our trading partners. There's many places to receive resources and updates on the continuing outbreak of H5N1 in dairy cattle, and those would be available after this cast webinar. And with that, I thank you for the opportunity to walk through what's been a pretty long but short 90 days so far in the dairy sector.
Thank you, Dr. Jonker. Yeah, I can't believe it's only been a few months. We've learned a lot, and we still have a lot to learn. Next up, we have Dr. Gauthier with USDA. And if you all haven't been doing it, we've been getting a lot of great questions. We're going to try to answer some of them live. If we don't get to all the questions, we are going to try to write those out and email them out to everybody who registered for the webinar. So continue adding your questions to the Q&A. Thanks.
Thanks very much. And my preceding speakers have given a really great summary of the outbreak in dairy cattle. So I'm going to focus on a couple of new things that we've been working on and give some updates about what we've been learning about the epidemiology of this infection, about the outbreak, and then to talk a bit about our voluntary herd status program. So we've been sending epidemiology strike teams out to 3 different states at this point to take in-depth interviews with any producer who'd be willing to sit down and complete some pretty extensive questionnaires. So out of, well, a little less than half of the herds have been participating in these epi surveys. And what we've found is that we've got a lot of risk factors that we could associate with moving the virus from place to place. We don't have particular smoking guns. We can't say this is exactly how the virus made its way onto a premises, but we can take a look at those risky activities that are happening on farm to get an idea of how this might be spreading around. And many of the farms reported, about a third of them, that they had received cattle within 30 days of clinical signs starting on those farms. And it was not unusual for milking cows, lactating dairy cows, to be moved from farm to farm. 51% of those, half of those movements involved trucks or trailers that were shared with other farms. And if you take a look at the graphic, they weren't cleaned and disinfected regularly. About half of those might be cleaned before using them again to transport cattle. So that's another way the virus can certainly move from one place to another. Shared equipment was a common finding, as well as shared workers. So workers would go from dairy to dairy. They were also working at dairies and poultry premises as well, which is another a really good way to carry that virus on our hands, our clothing, our shoes, and bring that to a new farm. So our key messages that we're learning from our EPI surveys is that the spread of H5N1 between the states is really linked to cattle movements of lactating dairy cattle. And then we see further local spread between dairy farms in an area in some of the states by movements of people, equipment, and vehicles, more than likely. So it's multifactorial. That usually can't pinpoint one exact activity or thing that caused the virus to be introduced, but we're certainly seeing lots of risk factors. Biosecurity really is the way that we're going to stop this, and that we can mitigate that risk of disease spread. And then everything that we've seen coming from FSIS and from FDA shows that our milk and beef supply are safe. Pasteurization works very well. And this is not a problem for the meat supply. So our recommendations, biosecurity really is the key. We can implement those biosecurity measures that are part of the secure milk supply plan. That existing plan really helps in this new case, in this new outbreak. Testing, as you heard previously, testing before moving cattle and minimizing the movement of cattle. We understand that we have to move cattle at times, but we can minimize the movement and reduce some of the risk of moving the virus from one state to another. We need to report if we're seeing cattle that are showing signs that could be avian influenza, and we need to conduct an investigation. Our state and federal animal health authorities are ready to do that, to investigate these cases and determine whether or not the herd is infected. We need to take a look at the interconnections between these farms, sharing workers, workers living in the same household and maybe working at different dairies. That's a risk. And identify as many of these connections as possible so that we can determine the ways that this virus is making its way from farm to farm. So a little bit about our response. You heard about the supports that were being provided. Those supports are available to both unaffected and affected producers. We do have a federal order in place that requires notification if a laboratory finds a positive test for H5N1 in dairy cattle. And we've also implemented a herd status program that was rolled out in early June. And in just a couple of weeks, we've signed up 4 herds in 4 different states. I'll talk a little bit about that herd status program. So these herds that need to move cattle interstate can really benefit from joining the program. And that if they test weekly their bulk tank milk, and they get a negative test back, then they can qualify for the program. And with weekly testing after that, they can move whatever cattle that they'd like to on a regular basis and not be required to do that individual animal testing that's required of other herds. The samples must be tested at one of our NOLM laboratories throughout the country. And so if they enter the program, test for 3 weeks in a row, test the milk, then they can establish an unaffected herd status. Then to maintain a monitored unaffected herd status, they would conduct weekly testing. And other than the health certificate, they wouldn't need any individual animal testing in order to move their cattle. If we find an affected herd, then that's something that we need to investigate and prevent movement of the animals unless they've tested negative. So next steps, enrolling herds in the herd status program, encouraging them to come on board. We expect that we'll see additional cases over the next few weeks as we do more testing. That's to be expected. We're hoping that the ELAP program, which is being rolled out soon, which will pay for milk loss associated with being an affected farm, will also encourage herds to test and figure out what their status is. USDA collaborates with our state animal health official partners, as well as the dairy industry and CDC, in order to identify cases, help herds practice good biosecurity, and prevent human illness associated with this disease. And I am looking forward to answering any of your questions.
Fantastic. Thank you for explaining a little bit of what APHIS has been doing behind the scenes and on scene. And we really appreciate everybody's help in this outbreak, because it's important to, as we've been seeing, the entire ag industry, not just one industry. We are a little bit over the top of the hour. We are going to stay an extra few minutes to answer some questions. So please hang with us, or you can check out the recording later. Chris, can we pop the slides back up?
Sure thing, Julia. Would you like to have the practical takeaways first, or would you just like
Yes.
to take the questions with everybody else? Okay, practical takeaways it is. Just 1 second here while I get it up for you. And I just want to say, before folks maybe hop off because you got another meeting to run to, I do want to definitely thank everyone that has been part of this. I appreciate the effort everybody's been putting in. It's very, very much appreciated. So, all right, just a moment here, Julia, and I'll get it up for you.
Well,
There
yep.
you are.
Yeah. So just some practical takeaways that people should take. And this, I think some of the questions will come into this too. We're constantly learning new aspects to this disease, what species are affected, what routes of transmission are happening. And the way that we have to share data. I think that's what a big lesson that we learned today, but adjusting our approaches as we get new information is really important. So what we know now was not the same as what we knew 2 weeks ago. And so this outbreak and response is going to be ever changing. Implementing biosecurity on the farm level is critically important. All of our presenters mentioned that, and we have some resources for some practical ways you can implement that on your farm, not just on dairy and poultry operations, but I think anybody who raises animals can take lessons from this on how to improve biosecurity to keep our animals from getting sick, not from avian influenza per se, but overall. Worker training is really important. We need to teach our workers and employees how to protect themselves, what they could be exposed to, provide those personal protective equipment, resources that are needed to protect human health. We want to protect our workers. That's really important. Food safety is critical. We already talked about how our meat supply and our milk supply is safe. But that comes to us as the consumer. We have a role to play in that also. So choosing those pasteurization products and then cooking meat to appropriate temperatures is really important. This communication from local to state to federal partners is absolutely important in an outbreak situation, but every day also. I think CAST has shown with this webinar, there's cooperation across the entire animal ag industry on figuring out this problem, but also working together, learning lessons from other industries. How do we protect how do we take those lessons and apply them to our own industry? The big thing is we know that different animals have those subclinical signs. You may not know that they're sick right away, but if you do notice abnormal signs in your animals, we really need producers to work with their local veterinarian. If you see sick animals, the quicker we can diagnose these, the faster we can get into preventive practices. So the next slide is hopefully you guys can take some screenshots of this. These are resources. Some of it Jamie already provided, but we have USDA, FDA resources, all the secure food supply plans, which are enhanced biosecurity practices that are specific to foreign animal disease or emerging infectious diseases like this outbreak in dairy cattle. There are some sector-specific everyday biosecurity education resources here, so depending on what animals you are raising. And then, as always, check with your state Department of Ag or your state vet's office or your local veterinarian to see what they recommend. But we'll stay on this slide for a minute, and we'll see if we can answer a few of these questions. So I'm going to go with Dr. Gauthier. We've been getting a lot of questions about transmission of the virus. And do we think fomites is one of those ways? So fomite direct contact with the milk. What are some other ways that, or other routes of transmission that are being investigated?
Yeah, we believe that people, vehicles and equipment really are moving this virus around. Anything contaminated with milk of infected cows can carry a large load of virus. We know that there's quite a bit of virus in the milk of infected cows. So anything contaminated by that is a potential fomite vehicle for that to get around. Our questionnaire involves everything from the presence of wild birds to wildlife, such as little mammals, rats and mice. We look at just about any pathway you can think of: water sources, feed sources, where the manure is going. But the thing that we're finding very common among affected farms is that they're sharing equipment, people, and vehicles.
Thank you. We have a few questions talking about oronasal transmission. Is that something that's being looked at right now?
It is. In the Agricultural Research Service, they are conducting studies where they've inoculated cattle to determine where the virus likes to hide and where the animals shed the virus from. And so it has been found in respiratory secretions, as well as a little bit in the urine. Manure doesn't seem to be a big place that we find a lot of virus, but milk certainly is.
And with that, that's a great transition to our next question. I'll put this over to Dr. Jonker. Pasteurization has been noted as a key safety measure. So what is the time and temp at which the live virus is destroyed or killed?
Yeah, so the work that's been done so far is looking at the time and temperature requirements in the pasteurized milk ordinance. So there is both high temperature short time as well as batch pasteurization. I anticipate FDA will be publishing some information here in the next couple of weeks on some pilot scale pasteurization research being done in coordination with the USDA Southeast ARS Regional Poultry Laboratory, looking at the time and temperature values that industry uses, which are typically actually a bit above what's in the pasteurized milk ordinance. There have been a few lab bench studies that have been done that demonstrate at minimum requirements of the PMO that it is highly effective at killing the virus. And so the PMO is really where you look at the minimum standards for what the time temperature requirements are.
Good information. Thank you. Thank you, Jamie. I'm going to pose this question to Dr. Culhane. I lost it. There we are. Any unique genetic changes of HPAI recovered from cats who died of HPAI on dairy farms?
Genetic changes, I'd have to refer to the virologist looking at those in particular. When we look at them at specific mutations, when we look at the cat viruses, they seem to be matching up 99 to 100% with the cattle viruses, and the cats viruses that are currently available are from cats that died on dairy farms with infected cattle. So they seem to be the same virus. For the cat, the virus goes to the brain. And it's a fatal infection for the cat in most cases. Thank you.
Those have been some sad cases to hear about. I'll pose one question to Dr. Gauthier again. We have a number of questions asking about non-lactating dairy cattle and if they play a role in transmission. And as a secondary question to that is, what about beef animals?
Haven't found it in beef animals so far, even in beef animals that live in fairly close proximity to dairy animals, which is interesting. So we're learning a lot about this virus every day, and we're finding that most of the transmission interstate has been associated with those lactating dairy cattle. That's not to say that it's not happened with other classes of dairy animals. So it is possible that non-lactating dairy cattle can move the virus from place to place. But that's the majority of our cases have been associated with the movement of lactating dairy cattle.
Excellent. And I'll ask the last question. I guess I'll open it up to everybody. It's a public health question because agriculture, we don't just work on caring for animals, we care for the people who care for the animals. And so how do we, since we know that people can get infected by this, what are some ways that we can help protect our workers? And moving forward, what are some lessons that you would take back to your industry?
I'll go ahead and start, Julia. When we look at the CDC recommendations of full PPE for workers, that's probably we're probably not able to implement that on farms. When you think about how the weather has been very recently in large portions of the country, being in full PPE, you start getting a potential worker safety issue of overheating. So you saw in what I put forth as to the big things that we're recommending, which is protecting the mucosa for those people that are in close contact with clinically infected animals. So we're focusing on the eyes, nose, and mouth as the place where we need to focus our efforts in the dairy sector based upon current knowledge and using that as the focal point. And there are reasons why we already recommend, for example, the use of goggles in milking parlors, because there's teat dips and sanitizers in there. There are tails on animals that, which if they get in contact with the eyes can cause, eye lacerations. And so this is really just in some aspects reinforcing standard practices that we recommend today, but really focusing on things that can be implemented on dairy farms to protect our workers and family members that are potentially dealing with clinically infected animals.
Excellent points, Jamie.
Early detection really is important to protecting people. So if we can identify those affected herds early on, and then people can take enhanced precautions to prevent themselves from contacting milk from infected cows, then we'll be further ahead at protecting our workers.
Absolutely. And I think, again, this is something, these are different techniques that can be used when a high path isn't what we're worried about. I mean, there are a lot of different viruses and bacteria that people who work with animals on a daily basis are exposed to already. And so we're reinforcing, I really like that, we're reinforcing things that we should already be doing on a daily basis. We're coming up on an hour 15, so I am going to close out questions and pass it back over to Chris. Thank you all for joining us. Great questions. We are going to answer those and send out some information after the webinar, but I'll pass it over to Chris.
Thank you, Julia. And thank you to everyone who participated today, both as a panelist, an expert, and certainly as a participant as well. Julia made a very key point there. We are going to make these resources available to you, so be on the lookout for them. Everything that CAST produces is free of charge. So these are free resources. That is the slides, the recording, work through the question and answers. We certainly had lots of questions we didn't get answered, so be on the lookout for these resources coming your way. Before I leave, though, and before you leave, hopefully, just a few things. First of all, I want you to note that CAST is an organization that works across a lot of sciences: food, animal, and plants. So if you have an interest in some of the topics you see on the screen at this time, new papers that we've launched, upcoming papers on a variety of topics, we'd love for you to follow us, particularly on LinkedIn. You can look for myself, or you can look for CAST as an organization, the Council for Agricultural and Science and Technology, on LinkedIn, so that you can be fully aware of what is coming out from CAST. Of course, we'd love for you to become a cast member. You can scan that QR code at this time to learn more about us. And again, you can go to our website. You can reach out to us via social media. And I said earlier, LinkedIn is one of the key things that we're posting on. Also, you can contact us by phone or email. We'd love to hear from you. We'd love for you to be part of CAST in some way. Again, thank you everyone for joining today. Thank you for all of our experts participating in this wonderful webinar. Take care, everyone. Have a great day. Bye-bye.